Enzyme action. Woohoo! Enzymes are proteins with specific conformations. They are biological catalysts, which means they are involved in chemical reactions, and it increases the reaction rate in the human body. So an important distinction is that all enzymes are proteins, but not all proteins are enzymes. This is important because proteins serve other important functions in the body besides being enzymes. Enzymes are chiefly involved in many of the metabolic processes in the human body, such as this one, which is the hydrolysis of glucose. They affect these processes in a way that allows them to occur at an accelerated rate than they could before, and allows them to use less energy than they did before, which just increases the body's overall efficiency. So this graph models the energy of the reaction of the hydrolysis of glucose, and the blue line represents the uncatalyzed reaction, so without the enzyme, and the red line represents the catalyzed reaction, so that's with the enzyme. In this case, that'd be fructase, the enzyme that catalyzes this reaction. So the little hump in the beginning of the line will represent the activation energy. That's the energy to get the reaction started. And as you can see, the hump for the blue line, there, the activation energy for the blue line, is much greater than it is for the red line, indicating that there's a much greater activation energy required for the uncatalyzed reaction than there is for the catalyzed reaction. It's also important to note that the ending energy is still the same for both. So how does an enzyme lower the activation energy? So for a reaction to occur properly, the proper functional groups of both the molecules must interact in the right way. But sometimes this doesn't happen, the wrong functional groups interact, so the reaction doesn't occur. So what an enzyme does is it increases the probability that a reaction will occur by holding one of the substrates in place, in this case, and this makes it more likely that the correct functional group of this substrate will react with the correct functional group of the other substrate. And this lowers the activation energy because it takes less activation energy for the reaction to occur. In other cases, the enzyme may hold both the substrates in place, which will decrease the activation energy required to synthesize them into a single molecule. Or the enzyme might hold the substrates, or a single substrate, in place for the purpose of dividing it into two other molecules. Notice that in all the cases, the enzyme is unaffected. It is ready to accept the next set of substrates. Only the substrates themselves are changed. The region of an enzyme that interacts with the substrate is called the active site, but the enzyme may also possess other regions that can interact with other molecules. These regions which are not the active site are called allosteric site. There are some molecules known as inhibitors. Competitive inhibitors are molecules that are not the substrate, but they bond to the active site, which prevents the substrate from bonding to the active site. Other types of inhibitors include non-competitive inhibitors, which bond to another site on the enzyme, like an allosteric site. This changes the conformation of the enzyme in a way that also blocks the substrate from bonding to it. Inhibitors play an important role in the body because they are involved in feedback regulation.
Hey, we made this video. Thanks for watching. Bye.
Bye.